The first talk, our guest Erik Aurell from Royal Institute of Technology, Stockholm, Sweden. Statistical genetics in and out of quasi-linkage equilibrium. So please, 40 minutes. Professor Aurell.
Okay.
Invite you, please.
Thank you. I'm here. It's a great honor to be invited and to have the opportunity to participate. And I thank Professor Zaharodny and Larissa and Valery and all colleagues. I recognize several of you in the room from when I was there just a month ago. Just a question, do you want me to share the slides from my laptop, or do you prefer to share the slides on your side?
As you wish. As you wish, we can use this presentation that is already shared.
Okay, then maybe we can go back to the first page then, just to give the title. And then I will say, okay, so the title of my talk is Statistical Genetics in and out of quasi-linkage equilibrium. And it's also a title of a recent paper which we published in Reports of Progress in Physics this year. Next slide, please. Yeah, So the background of this talk is that there has been a revolution in biophysics and in computational biology, essentially in the last 5 years or even less. Here is an illustration from the DeepMind company, which is an affiliate or a spin-off from Google, that for the first time has, in practical terms, solved the protein structure problem. That means given, and it's not enough with one sequence of amino acids for a protein, you have to have a family of sequences, closely similar sequences or homologous sequences. And from this information, you can infer using the technique of deep learning, which is in artificial intelligence, the three-dimensional structure of the protein. And as you know, structure in biology means function. This has been a holy grail for biophysics for at least half a century. It's an amazing advance, which has many, many practical applications. Next slide, please. And as you can read, every discovery has a prehistory, and this one also, and it's also very clearly stated in the abstract of the paper from Andrew Senior and others, which is the AlphaFold team, now 3 years ago or a little bit more. And I will read those sentences. Considerable progress has recently, this means before 2020, been made by leveraging genetic information. It is possible to infer which amino acid residues are in contact by analyzing covariation in homologous sequences, which aids in the prediction of protein structure. So just to unpack these two sentences, or the last one. So amino acid residues, these are the side chains or the side parts of the amino acids. And to know which pairs of amino acids are in contact is, of course, a consequence, something that you can deduce from knowing the structure. It is a partial information on the structure, and it's useful and has been proven to be useful in analyzing the complete structure. Now, the message of this paper in Nature by the AlphaFold team is that they were able to go much further, but still they acknowledge that this was an important advance. And the table below is an illustration of the kind of covariation that you have in mind. Namely, there is a table of aligned protein sequences which are in a family and therefore similar. And in this table, you have highlighted two columns. This means two positions, two amino acids on the same position, but in different versions of the same protein. And as you can see, if you look, there is a clear correlation between the two of them. So this kind of correlation is what means by covariation. You have that the distribution of amino acids at two pairs of position is not independent, but is dependent. Next slide, please. Yeah, next slide, please. And this discovery has essentially been made inside the field of statistical physics, and we are actually several in the community who have been engaged in this work. And I here just name one. No, come back to the previous slide. There was not two of them. Sorry. Please go back to slide 4. Yeah, so I've highlighted only the contributions of Martin White et al. When he was the first author, and Marcos et al. When he was the last author, which are now respectively 14 and 12 years ago. And there is a nice collection by Sergei Ovchinnikov, who is I guess originally Russian, but anyway, an American researcher who has been also very active in the field, both as a postdoc and later. Last time I met him, he was at Harvard. So in this, you have there an illustration of one of the databases of homologous proteins, and which then contains the same information as on the previous slide, and also other color markings with other indications of the type of amino acids and so on. And from this kind of tables of these positions of the different amino acid at the different positions, you deduce a distribution of the Gibbs-Boltzmann type, which is the one that you have below. That's the one that the green line is pointing on. That means a probability distribution over words, X. This means words in the letters, which give the amino acids, including, you have to do that when applying to proteins, the gap variable. And you have a kind of energy function, which has a linear term, which is like an external field, HI as a function of XI. Since there are 20 amino acids and also one gap variable, you cannot just write it as an Ising variable. This is more like a Potts model. And there is a quadratic interaction, JIJ, which then is dependent on the amino acid at locus I, which is XI, and the amino acid at locus J, which is XJ. So there is a way, the methods which the abstract of the AlphaFold paper refers to are methods to deduce these kind of Gibbs-Boltzmann models or distributions from tables of homologous sequences. And the prediction is that if you have a large term Jij between two loci I and J, this is a good predictor that in the structure, the loci I and J are also close. And this turned out to be quite successful and was for many of us an important focus in the last 10 years. And it's also been was very widely adopted in the CASP tournament, which is then partially, of course, a Ukrainian endeavor. There are many Ukrainians who've been active in the CASP tournament of the prediction of protein structure. Next slide, please. So there is, of course, the fundamental and I think very important general problem in science is why do these AI methods, deep learning work? It's a fundamental problem. Many colleagues have tried, and as far as I understand, there is no real understanding of why they actually work and when they don't work. That means the whole field has still an empirical field to it. For the earlier methods, which then do not give equally detailed information, that means these methods which are about inferring Gibbs-Boltzmann distributions from data, there is, on the other hand, a way to understand why they work and when they work. Of course, it doesn't work always, and I will get to that in a moment. So the talk is about those cases after this introduction. I will talk about a theory which is in essence more than 50 years old, which is maybe appropriate in a meeting dedicated to another discovery also 50 years old. And I will say something about applications to biological data, other applications than learning protein structure from sequences, where, of course, also AlphaFold and deep learning methods of this type do not always work. They require typically a lot of training data, and this is not always available. So there is also a space for these earlier methods still, but to other applications than to find protein structure. Next slide, please. So statistical genetics is the name coined by two gentlemen who published a PNAS and a review of modern physics now a bit more than 10 years ago. Richard Neher, when he was then a postdoc of Boris Schreiman, who is in Santa Barbara. And in particular, the review of modern physics contains a lot of material, developing a theory discovered by the Japanese population geneticist Kimura already in 1965. So that is the theory, which is now more than 50 years old. And I will give introduce a little bit of terminology. So linkage equilibrium means in population genetics, in the science of theoretical biology, that distribution of alleles, that means genetic variants, over loci, this means different positions, are independent. And so that would mean two columns in the table which you saw before, which are completely independent. Linkage disequilibrium means that they are not independent, and this can be due to fitness, or it can be due to inheritance, or both. Yep, and typically there are some mixture of both. Now, the formal definition, a population is said to be in quasi-linkage equilibrium, and this term was introduced by Kimura, and I will also refer to it as the abbreviation. Yeah, if multi-genome distributions factorize and genome distributions are an exponential family with terms no higher than the fitness function. So that means, previous slide, back to previous slide if you like. Yeah, so this means that actually, this one, yes. So this means that actually you get what you want. That means you get that the actual distribution of sequences in the space of possible sequences has the Gibbs-Boltzmann form, provided that certain evolution is in a certain phase, the phase which was first discovered by Kimura. And if you now remember this formal definition, I will then, in the next two slides, give you the conditions identified first by Kimura a long time ago when this has happened. So next slide, please. So the kinds of evolutionary forces which were taken into account by Neher and Schreiman, and mostly also by Kimura, is, of course, Darwinian selection, that is the tendency to maximize fitness. Recombination, that means the mixing of genomes in one generation to produce the next generation, which in higher organisms we also refer to as sex. And then this is, of course, obligatory in mammals, but there are some birds where this is not obligatory, but mostly in birds it's also obligatory, and it's present in all forms of life, more or less. This is an evolutionary force which acts on pairs of individuals, while Darwinian selection acts on single individuals. Then there's also mutations, which is important to some extent, and genetic drift, which I will mostly skip today. That would be finite population effects. And there are also some things which have never been considered in this, well-known effects in evolution, which have not been considered in this theory. So spatial effects. That means I'm here only considering well-mixed population. And next slide, please. And then the Kimura-Neher-Schreiman equation for DCA terms in the QLE, and this is an outcome of a rather long calculation, in particular assuming that mutation is not very high. So there is an overall rate of recombination R. There is a particular term which describes how much recombination there is between the specific loci I and J, which is the Cij. And there is a pairwise contribution to fitness, Fij. And then it turns out that the distribution will be in the Gibbs-Boltzmann form with terms, energy-like terms, which are proportional to fitness, but which are not quite fitness themselves. So you have you should think of a Jij as being, in physical terms, energy in terms of kBt. So this is dimensionless, while fitness here is a rate of increase of a certain combination. So this is per unit time. And R, which is an overall rate of recombination, is also per unit time. So in that way, the right-hand side is also dimensionless. So the theory which was derived is that in a particular limit, when the fastest process is the recombination rate, and it particularly faster than the rate of natural selection, which is given by the fitness function, which you here have the epistatic, that means the contribution from two loci, Fij, and also faster than mutations. And then you have this prediction, and this prediction can be tested in numerical simulation and can be used then as a predictor if you can infer the Jij from the data, which is what we did in proteins for the protein problem. This is also a prediction of something which is proportional to the fitness combination Fij. Next slide, please. And so here's one of those tests. Apparently, nobody else tested this before. There's no test of this kind also in any of the papers previously. But in a kind of an operational setting, well, why don't one starts with a population? One gives some epistatic fitness contribution here given on the x-axis as Fij true. And then one simulates a population of some number of individuals, in this case 200 individuals, of some genome. Here the genome was not very large, 25 loci, and you simulate for some time, and you extract the statistics at the end. That means you extract the different sequences that you have. And from those sequences, you apply the procedure of inferring the Gibbs-Boltzmann distribution, which according to the theory, according to the QLE theory, the Kimura-Neer-Schreiman theory, should describe the population. And so you get the Jij from this procedure. It's well documented in the literature. We have also all the references in our papers, and you can find more specialized reviews in the literature. So you use the formula on the previous slide, which is also on this slide, namely what you infer for fitness, Fij with the star, is two parameters, which I will skip how we, I will brush over those ones, but we have the Jij star, which is derived from the sequences. So Jij star is plugged in, this small equation gives you Fij star, and you can compare Fij star, which is on the y-axis on the figure against Fij, the true underlying one. And you can do many of these plots, and when things work, they more or less look like this. That means they look around the diagonal. So evidently, this is also an indication that the QLE is a test that the QLE theory works in numerical simulation, and a simpler test in many ways than many of them which were described earlier. So let me skip next slide. So let's go one slide and then one more. Yeah. So after describing this phase QLE, which I believe and I believe still deserves to be more studied and better known to, for instance, to physicists interested in the problems of evolution, let me discuss a little bit of things which we did on the loss of QLE, both earlier work by others and by us. Here's also the reference to our recent review, which you can find also on arXiv. Next slide, please. So the most important part of the theory, which is available in the literature, was also done by Neer and Schreiman, and later together with Marc Mézard, who is now at the Bocconi University in Milan, but used to be at the École Normale, the same as our next speaker, Massimo Vergasola. So there's this theoretical paper in 2013. And for the case when there is practically no mutation. I should maybe open a parenthesis that no mutation is a problem when you do this numerical studies, which I can get into in a more specialized discussion if anybody's interested. But I think one of the reasons there hasn't been so many tests of this theory in the easy way, which we worked out, is that the limit of new mutation in a finite and necessarily not very large population, which is what you can study numerically, is actually a bit delicate. Now, in this case, when you have practically no mutation, there is a transition in this overall parameter recognition rate from being very small. And in very small, you have a phase of clonal competition. That means you have one or several clones of closely similar genomes, which are derived by inheritance. So there's little recombination, which means most of the individuals in one generation are derived from only one individual in the previous generation, with maybe some small exchange of genetic material in between them. So you should think in this case maybe more of the kind of recombination in sex you have in bacteria, where or as in contrast to higher organisms, where they typically exchange at the time not so much. And maybe here they exchange not so much and not so often, and then you have clones of similar bacteria or viruses or whatever the organisms they are related by descent, and they compete. One of them is larger than the other one if it's more fit in total, and it then eat up the other one until there comes a new one eventually. And of course, the problem comes a new one is problematic when there's very little mutation. Then when R is very large, you have instead this phase of quasi-linkage equilibrium. So there is, for a finite n, there is a transition from one phase at small r where it doesn't happen, and to a phase at a large n. And there is a prediction in the literature of where this thing happens, namely, it is at a value of recombination rate which is proportional to the average epistatic fitness, that is the sigma, and then the square root of the logarithm of the population. So at an infinite population, this theory predicts, in fact, no QLE. However, it's not really a problem since you insert a very large number for n, and still, if you take the logarithm and the square root, you get a not very large number. For instance, if you put in Avogadro, the square root of the logarithm of Avogadro number is less than 10. Next slide, please. And what we discovered here is that if we do not have, if we have instead large or at least significant mutation rate, the loss of QLE happens in a qualitatively different way. And we have dubbed this non-random coexistence. So there is for finite populations, there appears an emittance regime fluctuating between something which is QLE and a non-random coexistence where you have here you have the top slide to the left is the frequency in the population at two particular loci in a randomly selected model. And you can see that there is the phase in time when they are both more or less at the frequency is around 0, or the magnetization is around 0. This is the QLE phase. Then intermittently there comes out another phase where it so happens that at one of these loci, the frequency goes up, and so magnetization goes to something like 0.3, and the other one, it goes down. So it goes to about minus 0.2. And they split away from 0 at the same time and come back to 0 at the same time, but they do not go to the same value. But this is, of course, part of the defined by these random couplings in this particular model chosen for illustration. You could also see that the total mean fitness in the population fluctuates, and so it is lower here for the QLE phase, and there's a higher mean fitness in the phase when these are when you go off from the equal, when you have a bias of certain loci. So in the random coexistence phase is in this case has higher fitness. You can also see a snapshot of the fitness distribution in one of these time intervals. It's in one in the middle, more or less, when you have a NRC phase. And you can find that the background in this is that there still is a bulk of the population which behaves as more or less QLE. That means you have a roughly Gaussian distribution of fitness, which is the left peak that you see on the bottom. And then there is a high peak of fitness, which comes in this particular chosen values at something around 4 on the x-axis, which is more or less then like a so the prediction is more or less that you have a phase when you have a background which is QLE-like, and then there is another part which is a number of clones, which are not exact clones because here mutation is larger, but they are kind of hazy clones, and there's the mixed up of no hazy clones to some hazy clones. And I think I have another 15 minutes, more or less. Is this correct? Let's see. Yeah.
Yes.
Yes, yes.
Yeah, about 15 minutes, right? So this is the phenomena which we discovered and which is presented in this one. And I have a couple of, maybe you can go to the next slide. Next slide, please. Yeah. And in the interest then of 15 minutes, let me say that we have a theoretical, a partial understanding of the transition and how it happens. We investigated, for instance, the finite end dependence, and there is a different end dependence in the transition rates from the QLE phase to the NRC phase, and then back from the NRC phase to the QLE phase. And one of them, in this case, the blue one, the waiting time goes up with the size, more or less exponential, which is more or less like Miller ratchet. And the other side, which is the QLE to the NRC transition in a finite population, this goes down with the size. That means the larger the population, the more likely it is for the QLE phase to decay to the NRC phase. And this waiting time is roughly inversely proportional to the size of the population. Next slide, please. And if you point this out at a certain phase diagram, we have more or less this kind of picture here, as you can see. Yeah. Next slide, please. So we have this, and then we looked first after possible manifestations of this kind of behavior, and we were in touch with Professors Good and Desai, who worked together with Lenski on analysis of one of the many analyses which have been made on the famous long-term evolution experiments, which Lenski started very long ago. These are those experiments where you have propagated 12 different, originally identical, but eventually over time different strains of bacteria in identical media, which are changed every day. And you can then follow the behavior of these 12 different test tubes, the population in those 12 different test tubes over now something also like 50 years. So this is another one of these 50 years evolution. In the beginning, not so much happened, and later more and more things have happened. This has been published in many times. And here we just focused on one of the many things, which is the appearance after quite a large number of populations. This, of course, was not visible originally, but later, after sequencing became possible, you could sequence samples taken from these populations at different time points. You can do these population-wide sequences, so you can have the whole meta-sequence landscape. That means the distribution of individuals in this population, the distribution over loci, for instance, at time. And you can see that in some of these cases, there is this stable long-term coexistence, quasi-stable coexistence, which has been found in, as per this paper in 2017, it had been found in 9 out of the 12 long-term evolution experiment populations. So we have a speculation that maybe this non-random coexistence could be a new and a possible explanation of these long-term, this behavior, which is a bit puzzling otherwise from the population genetics point of view. Next slide, please. So I'll end by saying a little bit about some applications. There are also some applications here which are more applications of the techniques of finding Gibbs-Boltzmann distributions from data. Proteins is not the only success story. There's a well-known series of papers by Chakraborty and co-workers on applying it to data on HIV. That means the sequences in one or several of the proteins visible on the outside of this virus when it's packaged. This is one reference. There are many more references. There's been interesting work by, again, Martin White and collaborators on RNA structure. We worked for a while, and also others worked on trying to find epistatis in bacterial pathogens. So it turned out that there was an interesting data set of about some thousands of the whole genome sample of the pneumococcus, which is a bacterium which gives rise to pneumonia. It's an important health. It's a disease-causing agent of considerable importance historically in the West and still today in the developing world. And so we published one paper on this in 2017, and there was a later paper by Debbie Marks from Debbie Marks' lab on the gonococcus, which is of course also an important pathogen all over the world. I will say something about the work that we did and others did on data from SARS-CoV-2, partially because, of course, the pandemic was very important, impacted much activity in all over the world, even the other pathogens which are kind of behind here, the gonococcus and the pneumococcus, over a longer time period have led to at least as much human suffering. But for the immediate impact, SARS-CoV-2 was larger, but also to highlight a very unique and very interesting data source, which is available for anybody to use in SARS-CoV-2, and which is much more impressive and richer than anything available elsewhere. And I will end by saying a little bit about a particularly nice application of this one by Martin White and collaborators, published last year. Next slide, please. So there is this fantastic data source called GISAID, Global Initiative on Sharing Avian Influenza Data, was the original meaning. Now it's more like a word by itself. And it became the leading repository for SARS-CoV-2 genomic data. And it holds, as of today, tens of millions of high-quality full-length genomes. So, only 6 years ago, we were able to publish on an important pathogen having some thousands of genomes. And this is now something that you can give to a master's student or even to a bachelor student to do a project on, because, much, much more data, because you can easily, with any selecting in any way that you're convenient to you, the number of data that you can have on SARS-CoV-2 is much, much more. Next slide, please. Yeah, so we were able to, yes. we were able to took this data. We started actually in 2020 in the first spring of the pandemic, basically with the idea that we have this technology which we have developed of finding Gibbs-Boltzmann terms from data, and we have the theory which we have worked on, which is relating those Gibbs-Boltzmann terms to fitness. So we have a way of predicting fitness and epistatic contributions to fitness from data. And maybe this could be of interest in the pandemic. That was the kind of motivation. Was also another paper which was published in the year afterwards, but which appeared on the biological archive, the bioarchive, more or less at the same time. And we were very happy about the second paper because we predicted many of the same interactions. If you see the circle diagram gives the it's one of those diagrams which contains many data. So there is I'm sorry. It gives an internal label of all the positions of the genome from 1 to 30,000, and these are the genomic positions on the SARS-CoV-2 data. Then there's an outer ring, which is an illustration of the different genes and the different proteins coding by those genes in SARS-CoV-2, which many of them have names. All of them have names. Their names are, some of them spike, been much in the literature. It's very important for the entry of the virus into its targets. But all of the others are also interesting for one another reason. And the illustration of the lines that you have there in the inside the black circle, these are predicted epistatic interactions between variations at different points. And those are the ones that one could get stably from, in this case, some 50,000 sequences for us, which was already an order of magnitude and more than had been available in any such study previously. And then Creswell, Clay, and Purry found very much the same. And also there were some differences in how we sorted out the confounding factor of inheritance. We did it in two very different ways, but still there was a great similarity between the two lists, and they had used more like 130,000. This was already in 2020. And now one can redo this on a monthly or weekly basis and can get much more. One can get time series of these predictions. Turns out that there is a very important time domain in this, which I will skip for today, but I hope I can talk about this some other time. I would like to highlight also this very nice paper from Martin White's lab, Rodriguez Rivas, where they started from a collection of the genomes of all the related viruses, the other coronaviruses, not SARS-CoV-2 only, but the others which were known to science before the pandemic. And then from this, they extracted predictions which they could verify on the same GISAID data. Next slide, please. And I think this is maybe the last slide. And this is. There's also a bit of non-random coexistence probably on this virus, which you can find if you plot the. This is an example of what you can find in up to August 2020. So these are now millions of sequences in total, and what you see here is per month or per week. I think this is per month. The frequencies of all the frequencies of the wild type, that means the original version of the genome, the original meaning in the first sequenced example, which is usually referred to as Wuhan Hu 1, because it came from Wuhan, and Hu was the name of the first author. And then you plot these frequencies, which are between 0 and 1 for all the 30,000 positions. And this is something which you can do now easily on a week-by-week basis. And you can get these pictures like this, which apart from that they are rather pretty, they show you that there is much dynamics inside these ones. You have most of them, of course, are frozen. They don't change. These are the frozen loci, and there you have lines which are plotted on top of each other. Some of them are clearly things going back up and down together, and they are related to they are variations which appear in the large clones, which are the variants of concern, which have these names of the variants of the virus, the alpha, the delta, the omicron, and so on. And then also you have something like an NRC phase. That means you have things which remain at intermediate frequencies over a long time. Turns out that most of those, I think almost all of those, are in the 5' or the 3' end of the SARS-CoV-2 genome. That means they are not inside any protein-coding gene. And it's known that there is a lot of recombination going on in SARS-CoV-2 viruses in general, and perhaps especially towards these age, depending on how the replication actually happens. And now, next slide, please, which might be my thank you slide. Yeah, I thank you for this possibility to present this work. I would be very happy to present it in more detail in another context, and there's lots more that can be done. Either another Zoom call, another Zoom conference, or hopefully my next visit to the beautiful city of Kiev. And my thanks to also the collaborators. That's Hongli Zheng, my former student, now in Nanjing, and Vito Dicchio, also my former student, and on the master's level though. And some theory was done together with Simona Cocco and Remy Monasson in Paris, and some earlier theory with Angelo Vulpiani and Hajun Zhu, Fabio Cecconi and Chenji Gao. And thanks to others funding and colleagues. Thanks a lot.
Thank you, Professor Aurell, for your lecture. Any question, please?
Erik, thank you very much. Can you hear me?
Yeah, I hear you very well.
Okay, thank you very much for your talk. And I have a question about, you have this rate of mutations. You speak about spontaneous mutations, but there are also, do you distinguish spontaneous and induced, for instance, by radiation? And the second question is, do you know about some studies on epistasis in Chernobyl area? There is really a very high level of mutations in wild animals, birds, plants, and so on.
Okay, thank you. That's two very different questions. Let me ask the first one, the second one first. So, of course, it's well known that Chernobyl has, after the nuclear accident, a higher mutation rate and many interesting features. But I don't know anything, more than what I and, of course, many others have read in the general press and general scientific press. It seems like Massimo has a question or maybe a comment on this. I don't know. I would love to hear. About mutations, well, there are, we only talk about here spontaneous mutations. There are many, how should I put this? There are many ways in one can make these models more detailed, more realistic, and more complex. Usually there's not enough data to constrain the models. In mutations, there is, for instance, the apart from including also induced mutations, as you say, there is also the well-known effect that you have mutation hotspots and in natural evolution. So mutation is actually not acting in the same way everywhere. on a genome. It doesn't act necessarily in the same way going forward and backward. And recombination is the same. There is not the same rate of recombination everywhere. Beyond that, you have the factor Cij, which comes into the theory. There's also an underlying recombination effect, but that recombination is not equally likely everywhere. So in all of these directions, which goes in the same way of saying that we don't include spatial effects, we don't include these effects on the space of genome, because there's a limited amount one can do immediately, and there's often not enough data to constrain the models in these directions. So those are both open directions, I would say.
Thank you.
Yes, the time to this lecture is over, so thank you very much.
There was a question from Massimo Vergasola, I'm sure. I saw it.
Yeah,
Or
there
there
was
was
a
a proof,
question.
maybe. Yeah.
No, I just wanted to know, you mentioned along the way that the limit of small mutation was problematic for technical reasons. Can you just briefly mention why?
Because if you have a finite population, after a while, then one clone takes over the whole population.
I see. Okay.
And the time it takes, the waiting time to go out of this clone when the distribution is frozen at one locus is very long.
So you essentially go to fixation. The problem is
Yeah, you go for fixation. And let's say if you have a 0 mutation, you go to fixation. If you have a very small mutation, you go to a quasi stationary state of almost fixation, and you have to wait an exponential time until you get out of fixation. Yeah.
And the way out is to average over many realizations.
The way out is to simulate with a bit of a higher mutation, or to be aware of that this is the problem, and then when you see to know how
But that's not quite the solution. You change problems. I mean, it's typical.
Okay, yes, that's true. No, but if you know that this is the problem, then you understand why the theory doesn't work, and you know where you have to. You can, of course, simulate for a longer time, but then you know why you have to simulate for a longer time. This is the difference. Well, I'm describing more or less the difference between our first paper on this, where we couldn't actually do this nice check of QLE, and the later paper, because in the first paper, which is the one, actually, you're looking now at my slides as a paper together with Fabio Cicconi, Chen Yigao, Angelo Vulpiani, and Haijun Zhu. We did quite a bit of theory, but we never got the simulations really to give what we wanted. And this was the reason. And we didn't quite understand that at the time.
Thank you.
Mm-hmm.
Thank you. Any question else? No, thank you. Thank you, Professor Aurell. We remember your previous lecture in our institute a few weeks ago. So thank you very much for your lecture.